I'm excited to speak with you today about HiFi sequencing with PacBio, and specifically our PureTarget long-read panels for targeted native DNA of challenging genes at scale. So I'll begin today by introducing PacBio's HiFi long-read sequencing technology and show the advantages of using HiFi for tandem repeat characterization. Then I will focus on the PureTarget repeat expansion panel and show you how it is scalable with an example for ataxia screening, how it is accurate through benchmarking of reference samples, how PureTarget is powerful enough to resolve complex repeats like RFC1, and how it is efficient with one multiplex assay able to replace up to 3 separate assays for FMR1 genotyping. Lastly, I will share some information about how we are expanding the PureTarget product line. HiFi sequencing provides long and accurate reads of native DNA. Sequence reads from 1 to 25 kb are possible, with high accuracy and few systematic errors. There is minimal bias, so you get the best coverage of the human genome of any sequencing technology. With native DNA molecules, you get methylation data with every standard library prep in the standard sequence files. Compared to short-read NGS, HiFi reads are up to 100 times longer and just as accurate. The reason HiFi sequences are so accurate is because they represent the consensus sequence of each library molecule. When making a sequence library, you start with a double-stranded template molecule. Then you ligate special hairpin adapters called smart bell adapters to the DNA, which results in a circular DNA library molecule. During sequencing, the DNA polymerase uses the library molecule as a template and traverses the circular molecule many times while we record the incorporation of fluorescently labeled bases. During sequencing, the DNA polymerase, finally, the long polymerase read is processed into subreads, which are shown here as yellow and purple. And then a single consensus sequence, a HiFi read in green, is computed. All of this processing is done on board the sequencer, so the output of the run is simply high accuracy, long HiFi reads. PacBio workflows are familiar to anyone doing NGS, and we have products to support you from sample prep through analysis. Our automation-compatible nanobind kits for DNA extraction support high-quality DNA, perfect for long reads, from a variety of sample types, including blood and saliva. Our library prep kits support whole genome, targeted DNA, and full-length RNA preps, and can be run manually or with automation. We have multiple sequencers on the market, including the new Vega benchtop, which is easy to use and affordably priced, and our mid-throughput Revio system for labs running at higher scale. Lastly, our sequencers use advanced algorithms from Google Health, run on the latest NVIDIA GPU processors, so we can deliver sequence data to you that is both powerful and in conveniently sized files. The Revio and the Vega system are PacBio's 6th and 7th generation sequencers. We launched our first instruments 15 years ago in 2011, and since then have made steady progress and dramatic improvements in throughput. To sequence a human genome on our early long-read sequencers required hundreds or tens of SMRT cells. Now with Revio, you can generate 40x coverage of a human genome on each SMRT cell with a standard WGS library. And each Vega SMRT cell produces 20x human WGS data. We have also improved our data quality, enabling HiFi sequencing, the consensus sequencing, directly off instrument, starting with 2020 and our Sequel IIe system. For human genomics, HiFi sequencing enables you to access more of the human genome. In 2022, a team of researchers led by Sergey Nurk at the National Human Genomics Research Institute in Washington, D.C., finally published a telomere-to-telomere version of the human genome. In this paper, they assessed the ability of different sequencing technologies to accurately map to the new human T to T reference genome. They found that more than 99% of the human genome is mappable and thus accessible to HiFi reads. Illumina falls short, and their data is not able to map to 200 megabases of the genome, which is equivalent to the length of human chromosome 3. If you can't map to the reference, you can't call variants. What does this improved mappability and HiFi read quality mean for human variant calling? Here I'm showing variant calling accuracy as measured by F1 score across different coverage levels for HiFi in pink compared to 35x Illumina and 60x Oxford Nanopore ONT. On the left, we see that PacBio beats Illumina and ONT at SNPs at about 20x coverage. HiFi does perform worse than Illumina for indels, but much better than ONT, even at 60x coverage for Nanopore. For structural variants on the right, long reads in general are much better than short reads for structural variant calls, and HiFi has better accuracy than higher depth ONT, starting at about 15x HiFi coverage. We don't have time to go into a lot of detail about this analysis, but you can pull up an app note with this QR code and learn more about this work. Altogether, HiFi can give you a reference quality complete genome and variant calling at all known variant classes, including at more than 400 medically relevant genes that are in the so-called dark regions because they are inaccessible to short reads. This new class of WGS includes, of course, SNPs and small indels that you can get with short reads, but also structural variants, methylation calling, tandem repeats, segmental duplications, and then, of course, phasing and haplotyping with the long reads. So let's talk more about tandem repeats. Repeat expansion disorders are relatively common, with conservative estimates of 1 in 3,000 individuals impacted by these diseases. There are about 60 disorders caused by expansions in short tandem repeats. When we look at the rate at which these disorders have been discovered and lay on the predominant technology used at the time, we see a large acceleration in discovery during the long-read sequencing era. In fact, more than 50% of disorders have been discovered in the last 10 years. Recent work analyzing 16 repeat expansions in more than 80,000 genomes from 2 large medical genomics cohorts, the 100,000 Genome Project from the NHS in the UK and TopMed from NIH in the US. They found repeat expansion mutations were 10 times more common than previous estimates, impacting more than 1 in 300 individuals. The disease incidence for repeat expansion disorders is lower than this, so either disorders are undiagnosed or have incomplete penetrance. Interestingly, in this study, they also found that most repeats are present in all broadly defined human populations, although the frequencies can vary. So for example, in the East Asian population, we see repeat expansions in DMPK and C9orf, which are common in European populations, are more rare in East Asians, whereas Notch2-NLC and TBP and ATN1 are relatively common in East Asians. As I'm sure many of you know, the length at which a repeat becomes pathogenic and causes disease depends on the gene and the disease. Here's a summary of 35 genes that cause repeat expansion diseases. In black, we see the repeat length range for healthy individuals, and in pink, the length of the pathogenic repeat size. You'll notice that for some diseases like Fragile X, the pathogenic range is much longer than it is for other repeats like SCA17. Many long repeats like C9orf, RFC1, DMPK, FMR1, FXN are so long that they're not accessible to short read sequencing technology because the length cannot be accurately determined with short read sequencing. A retrospective study by Genomics England in 2022 using short-read sequencing showed this. The authors explicitly noted that 4 of the 13 repeats that they studied, including FMR1, may have their repeat expansion sizes underestimated by short reads. Because of these limitations of short reads, many diagnostic labs continue to run classical, non-sequencing-based genotyping assays for repeat expansions. 2 examples of such assays are shown here: repeat prime PCR and Southern blots. Both of these assays have limitations. They're single gene assays, and RP-PCR has limited size resolution and no methylation information, whereas Southern blots are very labor intensive and they often require the use of radioactivity. In fact, if we look across a number of classical genotyping methods, we see similar limitations. This table is based on a recent review by Depien and colleagues. Here we're comparing Southern blot, fragment analysis with capillary electrophoresis, repeat prime PCR, and long-range PCR. And we evaluate the technology on the information obtained and how easy and scalable and cost-effective the method is. For repeat size, Southern blots and long-range PCR do quite well resolving long repeats, but the other methods can't resolve repeats longer than about 800 bases. None of these approaches can resolve repeat sequence in an unbiased manner, and the ease of running these ranges from difficult in Southern blot to easy for capillary electrophoresis. None of the methods can multiplex many targets, but the PCR-based ones can multiplex samples. Cost is low for individual targets, except for the Southern blots. And then I'll note some additional limitations, such that repeat prime PCR is reliant on pre-ascertained repeat motifs, and long-range PCR has the biases associated with any PCR assay, size bias, and issues with high GC regions. Now let's look at why HiFi read length is so important for repeat expansion genotyping accuracy. Here is a swim lane plot summarizing data for over 100 samples at 16 different repeat expansion loci that were sequenced with HiFi. Each row shows the size range of the different pathogenic categories. So the blue section is the range for normal alleles, yellow is for pre-mutation or intermediate sized repeats, and orange is the pathogenic size range. The data points represent the repeat length determined from HiFi sequencing. We expect the data points to fall within their corresponding swim lane if our genotyping and our expectations of pathogenicity in the sample are accurate. Below the swim lane plot, I've added the length of a short read and a HiFi read. Clearly, you can see that HiFi reads which can reach lengths longer than 20 kb, are able to sequence through the full pathogenic size range of all of these repeats and more accurately size the repeats, compared to short reads, which are typically 150 base pairs and are not able to accurately size pathogenic ranges for many of these targets. PacBio has developed a software suite that is designed to provide a best-in-class repeat expansion genotyping and detection. The tool is named Tandem Repeat Genotyping Tool, or Target. It takes as input aligned HiFi reads and a repeat definition file, which lists the motifs, the coordinates in the genome, and the expected repeat structure. It generates a consensus sequence for each repeat allele between unique flanking sequences shown here in green. It also determines the methylation status of each CPG site in the repeat as well as the flanking sequence. And it determines the motif boundaries of the repeats. The output of Target is a target plot, which is a visual which shows alignments of all of the reads in the 2 allele categories and the structure of the repeat. And it also produces a VCF file, which gives you motif counts, allele lengths, read depths, and other basic stats. Now that we've introduced HiFi sequencing and its advantages for repeat expansion genotyping, let's dig into more details about the PureTarget repeat expansion panel. PureTarget repeat expansion panel is part of an end-to-end PacBio solution for comprehensive characterization of repeat expansions. The workflow starts with high-quality DNA extracted with PacBio's nanobind kits. Although we and our customers have tested many other extraction kits with PureTarget, so you can pull up an app note linked to this QR code to see a list of all of the different extraction kits that we have tested. The officially supported sample types are nanobind extracted human blood using our RBC lysis protocol and lymphoblastoid cell lines. We see best performance with these types of samples. The PureTarget kit does target enrichment and library prep for a panel of 20 clinically relevant repeat expansion targets. Samples are barcoded with our SmartBell adapter index plates, and the libraries can be run on all of our HiFi systems. You can multiplex up to 48 samples on Revio and on Vega, and 24 samples on Sequel II. Our analysis software in SmartLink uses the Tandem repeat genotyping tool, or Target, which I just described, to return genotypes in the GUI software. It also produces library QC plots like coverage plots and coverage statistics. Here are the 20 clinically relevant targets included in the off-the-shelf PureTarget repeat expansion panel. We have 10 targets associated with spinocerebellar ataxia. We also include RFC1, FMR1, HTT for Huntington's, AR for spinal bulbar muscular atrophy, and several other targets. In addition, it is possible to design and order custom guides and add them to this panel, and we have customers that have successfully done that. A little bit about the library prep. So PureTarget is a PCR-free library method, and it uses CRISPR-Cas9. As I said, we recommend starting with high molecular weight DNA extracted with our nanobind kits. We see best performance when 50% of the fragments or more are longer than 30 kb. The input requirements are as low as 1 microgram of DNA, although if you want higher coverage, you can increase up to 4 micrograms. The first step of processing the high molecular weight DNA is to dephosphorylate and block the ends of the DNA. Next, we have a pool of guide RNAs and the Cas9 enzyme, which will cut on either side of the targets of interest. There's one cut site on either side. The length of normal alleles, library molecules, is about 4 to 5 kb, and an expanded repeat may be as long as 20 kb or even longer. After the Cas9 cutting, we do a DA tail step on the cut ends, and then we ligate those hairpin SmartBell adapters. The final step in the library is a nuclease digestion, which removes non-circular templates, such that you're left with an enriched library of the targets of interest. PureTarget has several advantages in terms of workflow. You can go from sample to answer in 3 days, with 2 hours for nanobind extraction, 8 to 12 hours for preparing 24 or 48 samples. We generally run 24-hour movies on any of the HiFi systems, and the analysis is very quick. It takes about 15 minutes on recommended compute resources. Compared to methods that use PCR, like PCR amplicons or hybrid capture methods, there are big advantages in terms of data quality for PureTarget. Of course, we get methylation because we're sequencing native DNA. There's no GC dropout because we're not using PCR. There's less size bias compared to PCR-based workflows, and we don't have any PCR-based replication errors, which is important in a tandem repeat region. Because we're not using PCR amplification and we need to cut out intact pieces of DNA around the repeat, you get better on-target coverage and with higher quality long fragments of DNA. So here's an example from a customer run where we're looking at coverage on the PureTarget panel versus the quality of the DNA, which is measured as GQN, or genomic quality number. So a GQN at 30 kb of 1, like the red sample, means that 10% of the fragments are longer than 30 kb, so not very many. Whereas a GQN of 7 means that 70% of the fragments of DNA are longer than 30 kb. We generally recommend DNA quality with GQN 5 or greater, and you can see that the samples that have that high quality have read depth or coverage of the PureTarget panel around 300 to 400x. Now let's see some examples of the 4 key aspects of repeat expansion genotyping that you can get with PureTarget. First, because HiFi reads are very accurate and they can sequence through the short tandem repeats, we can resolve the repeat sequence, including identifying interruption motifs like the AGG interruption, which is present in the FMR1 expansion. Because we're sequencing native DNA, we can call methylation. So here we're showing an expanded FMR1 repeat where each of the CPGs that are methylated are colored as red. And so we can see that this expanded allele is hypermethylated across its length. We can also accurately size even very long repeats. Here's an example for RFC1 where we're able to size very long repeats of about 6.5 kb. And because we get high depth with a targeted assay, we can profile mosaicism, meaning we can look at variation from cell to cell in the length of the repeat. And here's an example for the C9orf72 repeat expansion. So let's revisit this comparison table and add PureTarget on the right side so you can see all of the advantages at once. With PureTarget, we can resolve very long repeats. We get the sequence of the repeat, including those interruption sequences, like in the FMR1 example. The workflow, because we can multiplex samples and we can multiplex targets in a single prep, is easy to moderate. Again, multiplex targets, multiplex samples. This makes the labor cost quite low compared to single gene assays. And finally, we get methylation. With high depth, we get mosaicism. There's no GC dropout, and there's less size bias compared to PCR. So lastly, I just want to summarize the product specs for PureTarget. Our supported sample types are nanobine extracted human blood using the RBC lysis protocol and lymphoblastoid cell lines. The DNA input can range from 1 to 4 micrograms per sample. We recommend having GQN at 30 kb greater than 5, so 50% of your fragments are longer than 30 kb to give the best coverage. With these types of data quality specs, we typically see mean target coverage greater than 200x and min target coverage per target of 50-fold. You can multiplex 48 samples on Revio and Vega and 24 samples on Sequel II. The library size for the unexpanded alleles is about 4 to 5 kb, and expanded alleles can have library molecules that are 20 kb or longer. And of course, we detect methylation. So now that I've introduced the PureTarget product, I'm going to go through some examples, starting with how PureTarget is scalable and efficient for genotyping ataxia repeats. For diseases like ataxia, there are about a dozen or more than a dozen about a dozen genes that you need to screen. So PureTarget offers a scalable and a streamlined workflow where you can assay them all at once. In this example, a lab may be running repeat prime PCR and southern blots across a panel of different ataxia genes in multiplexes or in multiple samples at once. So with PureTarget, you can literally replace dozens of individual tests with a single long read panel. So there's a lot of labor efficiencies with this approach. And last week, we were really excited to see the announcement from Athena Diagnostics, which is a business of Quest Diagnostics, which is one of the largest testing labs in the United States. They recently announced that they are using PureTarget as part of their upgraded ataxia offering. One of the benefits that they explicitly call out in their flyer shown here, which was advertised at the ACMG conference in Los Angeles last week, they explicitly called out that the PureTarget approach gives them a streamlined workflow and it enables them to process larger sample pools. Now let's turn to accuracy, where I'll share some results of technical replicates where we benchmarked against reference samples. So our approach here is quite simple. The Coriell Institute has a bank of many samples with known repeat expansions. We prepared samples with PureTarget libraries, and then we sequenced them multiple times on each of the HiFi platforms, and then compared the results from run to run and against the expectations. So in this table, I'm showing technical replicates of 11 different reference samples with known expansions. We see the expected motif, the expected genotype, so the number of counts of that motif in the short and the long allele. And then we have the genotyping results on Revio, on Vega, and on Sequel2 for the short alleles and for the long alleles. We recently presented this data at a poster at ACMG, so if you want to learn more, you can pull up that poster using this QR code. For the short allele, all samples gave 100% concordance in the motif count across all three platforms. For the long allele, all samples were concordant within 10% of the motif count across platforms. And in every case, the interpretation of the motif count in terms of normal or pathogenic was the same. You may notice some small differences from expectations. So let's dig into one of these examples, ATN1. So here's a data set that we did on Vega. The expected genotype is 16 CAG repeats for the short allele and 68 for the long allele. When we genotyped this sample with Target, we counted 20 repeats in the short and 72 in the long. The short allele is clearly in the benign size range, and the long allele is in the pathogenic size range. So when we look at the sequence of the repeat shown here from the target plot, you can clearly see two alleles, the short and the long, with most of the repeat consisting of this blue CAG repeat, although there are two copies of this purple CAA motif in the beginning of the array. You can also see some mosaicism where there are some longer repeats in the long allele cluster. We've sorted the reads by repeat length, so this is what we call a waterfall plot. So when we count up the individual motifs CAG or CAA, we see that that sums to what we just saw, 20 and 72. However, it turns out that the expected genotype is based on an assay that only looks at contiguous runs of the CAG repeat. So these four repeats in the beginning, the CAG, CAA, CAG, CAA, is omitted from the classical genotype. And when we remove those four copies, which are not counted in the classical genotype, we get a concordant genotype. So the take home from this example is that the repeat definition matters and that some recalibration may be needed with a new technology like long reads relative to older classical methods. But we can easily explain these discrepancies because we get such high resolution look at the repeat sequence itself. Okay, so the take-home messages so far is that the PureTarget panel is scalable. You can screen 20 targets at once and up to 48 samples per library. And this scalability was what led Quest Diagnostics, one of the largest testing labs in the United States, to adopt PureTarget in their new Ataxia offering. The PureTarget method is accurate. Genotypes are reproducible in reference samples across our different sequencers and across technical replicates. And this approach is powerful. You may see small but consistent differences with classical genotypes, but these are easily explained using the fully resolved HiFi sequence of the repeat. So now I'll show you another example of the power of PureTarget for a very challenging target, RFC1. So RFC1 is associated with Canvas disease, which stands for cerebellar ataxia, neuropathy, and vestibular areflexia syndrome. It has autosomal recessive inheritance, and it's relatively common and observed in many global populations where it's been screened. The challenges associated with genotyping RFC1 are shown here. First, the repeats can be very long. Repeat motifs are about 5 or 6 bases long, so having an expansion of 2,700 repeats means the repeat itself is about 15 kb in length. There is very high diversity in the motifs present in RFC1. There's multiple motifs that are benign, and the most common pathogenic repeat is this orange one here, which is 2 A's, 3 G's. And there are a number of unknown, repeat motifs with unknown pathogenicity. Lastly, these repeats are very GA-rich, and GA-rich sequence can form unusual secondary structures in the DNA, which make them challenging to sequence. So here are 2 examples of a sample with a double pathogenic RFC1 expansion. The expected genotype of both samples through an RP-PCR assay was they were expecting a double expansion of that pathogenic orange 2A, 3G motif. When we sequenced this with PureTarget and genotyped it with Target, we indeed found confirmation of these double expansions. And the longest expansion that we sequenced was over 1,200 motifs, which was over 6 kb, and we could sequence all the way through this. Here's another example that is even more interesting. So this is an individual that was shared by one of our collaborators. Again, it was genotyped with RP-PCR and expected to have 11 of this blue motif, 4 A's and 1 G. And it was expected to have an expansion of these 2 different motifs, the purple and the orange. Yeah, okay, so when we sequenced this sample, we saw something a little bit different from the expectations. So the allele 1 actually matched the expectations. We found 9 copies of the blue motif, which is similar to the 11 that was expected. However, when we looked at the expanded allele, we didn't find the pathogenic orange motif. In fact, we found a long expansion of this yellow motif, which is 3 A's and 3 G's. And it turns out that the repeat prime PCR assay that was run in this lab is unable to distinguish between the orange and the yellow motifs. And so it's likely that this individual is not a carrier for the pathogenic expansion, which is a pretty interesting result. Okay, so the last example I want to share about PureTarget is the ability to consolidate multiple assays as an example for FMR1, which causes Fragile X. So when genotyping FMR1, there's three important aspects to this. First and foremost is the repeat size. And so you can see, and you probably know, that the FMR1 expansion can be quite long. And for this reason, and because it is a GC-rich repeat, it is very challenging to genotype with short reads and PCR-based methods. The second aspect of genotyping that's important are detection of interruption sequences. And that's important because the presence of interruption sequences stabilizes the repeat when it is transmitted from mother to offspring. This figure shows the probability of expansion from mother to child when there are 0, 1, or 2 AGG interruptions present in the repeat. The expansion probability is lower when there's more AGG interruptions present. So this is really important for carrier screening, especially for potential mothers that have pre-mutations. If they have an AGG interruption, the repeat is less likely to expand when they transmit to their offspring. Lastly, methylation is important for FMR1 because it's the mechanism of action for Fragile X. The presence of the CGG expansion results in hypermethylation of the promoter region and repression of gene expression. So it's common for methylation to be tested in children that are suspected of having intellectual disability caused by an FMR1 expansion. So with PureTarget, we can easily detect all three aspects in a single multiplexed assay. We can precisely size the repeat, shown here. We can identify the interruption sequences in the repeat, and we can call methylation in the expanded alleles. So here's a table of another Coriell reference sample showing the consistency of the repeat length size across 8 different technical replicates on different HiFi sequencers. So we have the Revio with the new Spark chemistry, Revio with the older non-Spark V1 chemistry, and then Vega. And you can see with the short allele, we have very completely consistent calls in terms of the length of the repeat in base pairs. And for the long allele, it's plus or minus 2 bases. So very consistent. And we get really nice coverage across all of these platforms. Okay, so the last two take-home summaries here. PureTarget is powerful, and the method can resolve complex repeat diversity at RFC1. And this assay is very efficient. A single multiplexed assay can replace 3 different assays for repeat size, interruption sequence, and methylation at FMR1. So with that, I will share a little bit about the future roadmap for PureTarget and make another plug for our sequencing instruments. So PureTarget has been on the market for about 1 year, and in that time, we've seen really enthusiastic adoption of the method for both research and diagnostics research. And we've received a lot of helpful feedback from customers, so we're able to expand the product line this year. Some of the things that we're focused on are scale. So we are working on higher multiplexing, up to 96 on Revio, as well as an automated library prep available for Revio. We are expanding the content, so we will be adding additional repeat expansions to the panel. And we are developing panels for other so-called dark genes, especially genes with paralogs and pseudogenes, which are relevant for carrier screening. We are also building out our capability with custom panels. While you can't necessarily order custom guides directly from PacBio, we are working with partners, and we will have recommendations for design tools and vendors that you can order custom designs and then spike them in or add them to the off-the-shelf panels. We're also focusing on performance, expanding our support to multiple and new sample types, including saliva. And we are working on improving the on-target coverage. So I'll leave you with a plug of our 2 on-market HiFi sequencers. The Vega benchtop is a perfect fit for PureTarget. It can give you HiFi sequencing within reach. It's a very affordable instrument, so definitely reach out to your local sales team and learn more about bringing this instrument into your lab. The data and the workflows on Vega are the same as what is available on Revio. It's a very easy to use sequencer, and it also can run all of the other applications that PacBio instruments can run, including full-length RNA, whole genome, and of course the PureTarget targeted panels. And then for labs running at a higher throughput, we have the Revio, which is a mid-throughput sequencer. Our new Spark chemistry enables you to load as little as 500 nanograms for a whole genome library, and you get about 120 gigabases or 40x of a human genome with a whole genome library. Revio allows you to do methylation as well as 6-MA calls with the FiberSeq assay, and because of the lower input amounts enabled by Spark, you can, the system is accessible to many, many more sample types. So I want to end by thanking our collaborators. We have some wonderful collaborators at the University of Athens that donated those RFC1 samples that I shared with you, and then my colleagues at PacBio who have worked on developing the assay and marketing the assay. And I believe we have plenty of time for questions, so I'm happy to take any questions.
Okay, so let's start the Q&A time, Sarah. So the first question is, I'm interested in some repeat expansion that on the panel, like FGF14 or Notch to NLC. Do you have plan to add those targets to the new panel?
Yes, actually we do. So you can reach out to the local team if you're interested in those two targets. I would love to speak with you and see if there's any other targets that you may like to add. We will be including those in the version 2 of the repeat expansion panel. But as I said, there is the ability to do some customization of the panels, and you can design and order guides from a vendor like IDT, and then add those to the panel and then follow the standard protocol from there. So I can share some information if you reach out to the local team, giving you some guidance about that. But those two targets that were asked about, FGF14 and Notch2-NLC, those will be on the version 2 repeat expansion panel, which is coming out in 2025.
Okay, and then there are some other panel questions. There are some targets on the panel that I am not interested in the sequencing. Can they be removed?
So I think as we see labs trying to kind of standardize their workflows, it's common with exome sequencing or whole genome sequencing to do in silico panels. So you sequence more, but you only report out what you're interested in. You can do something similar to that with PureTarget. So when you run the analysis, the analysis takes a bed file of a list of your targets. So if there are targets on the panel that you don't want data for, maybe Huntington's is something that you don't want to report out to a patient, you can remove that line from the bed file. And when you analyze the data, the reads are not output from the workflow. So that's a very zero cost way to customize the panel by making kind of an in silico or slimmer version of the panel.
Wait, there is another panel gene questions. So I'm interested in using PacBio for CYP21A2 and ATBA1 and ATBA2. Will you add that to the panel?
Yeah, these are what we call dark genes. So CYP21A2 has a pseudogene, I think, pseudogene or paralog. It has a very similar, I think it's a pseudogene, a very similar gene copy, and it's very challenging to genotype. So in fact, we do have designs for that target, and we are including it in the carrier panel. HbA1 and 2 this is an interesting target that has some large deletions, which can be challenging to genotype because of the homology between the two genes. And yes, these targets are on the carrier panel as well.
Another panel question. So I'm interested in the application of PacBio long-read sequencing to genotype FSHD. Is there the targeted repeat panel for the FSHD?
Yeah, this is a very, very challenging repeat. And we have looked into this for PureTarget, but the repeat sizes are actually too large to effectively target with PureTarget. However, we do have a new software tool for genotyping this target for whole genome data. So there was actually a talk about this by one of the members of the Comp Bio team at PacBio named Zhao Chen. She spoke about her new tool at the ACMG conference last week. So it won't work in the PureTarget panels, but it is something that can work with whole genome sequencing because the reads that you get with whole genome are just consistently longer with that approach.
Thank you. And there are several technical questions. So number one is that when would you recommend using PureTarget versus hybrid capture libraries for long-read sequencing?
Yeah, that's a great question. We do have partnerships with companies that do hybrid capture workflows, like Twist Bioscience has some targeted long read panels that work on PacBio. I would say if methylation is important to you, or if it's a repeat expansion where PCR can cause problems, the PureTarget approach is a better one to take. However, if you don't care about methylation, if the sequences that you're interested in don't have any sort of challenging contexts, the hybrid capture approach is a good one to take, especially if you have a very large panel that you want to sequence on HiFi. Right now, the ability of us to design large panels on PureTarget, meaning multi-megabase size panels, is limited, whereas you can work with Twist and give them a list of genes, and they can design the capture probes for you. We have two panels that we designed with Twist. One is a 2 megabase PGX panel, and another is a 20 megabase dark genes panel with 400 genes. So if you have a large panel, very large panel, multi-megabases, I would say hybrid capture is the way to go. If it's a smaller panel, you care about methylation, it's repeat expansion, PureTarget would be the way to go. And then the last thing I'll add there is that the workflow for PureTarget is actually easier and faster than hybrid capture. So if turnaround time is important to you, then PureTarget is a better option. Turnaround time and also automation. We have successfully automated PureTarget, whereas the Twist hybrid capture workflow has been challenging to automate.
So the next one is that, can I use the buccal swabs with the PureTarget?
Oh, buccal swabs. We have had some customers try buccal swabs, and it has not been successful. However, saliva is a sample type, a non-invasive sample type that does work with PureTarget. And PacBio has a protocol for nanobind extraction of saliva that is collected using the Origin tubes from DNA Genotek. We also have seen success with the QIAsymphony saliva extraction protocol with PureTarget. So buccal does not work, saliva does work.
Okay, then what do you think the benefit of nanobind CBB kit compared with other HMW DNA extraction kit?
Yeah, We have tested a number of other extraction kits. I think Nanobind and Monarch from NEB, they perform quite well. Monarch from NEB does not have automation, whereas the Nanobind kits do have automation, so that is an advantage. And, you know, if you are, there's a convenience factor of ordering your extraction kits from PacBio and the library prep kits. We have really, we have a very deep understanding of the nanobind extraction kits and how they perform with PureTarget. So it's just a very robust, well-studied, and a supported sample type that you can get good support from PacBio for.
And I'm not sure whether you can answer, but the question is that I was wondering the algorithm can determine the repeat count accurately, even when the expanded allele exceed about 25 kb.
Yep, that's a great question. There are some expansions that have very, very long repeats. Some of them include CNBP, which causes myotonic dystrophy 2, C9orf72, RFC1, I think FXN. Those can all be very long. One of the things that we've done with PureTarget is we don't just rely on the HiFi Q20 and above reads. We also include reads that may have lower quality, but still have both adapter sequences on either side. So sometimes for a very long repeat, the polymerase can't go around that molecule as many times because it's so long. And so we do retain some of those very long reads. They just may have lower quality. I don't know if you have a repeat that's 25, 30, 40 kb, whether you need to know the exact number of motifs. That may be a little bit more challenging because the read quality may be lower and you will have fewer reads at that length. But we have sequence reads that are more than 40 kb in length from some of these very large expansions.
Okay, so next question is that what kind of program do you recommend for design the sgRNA when I want to customize the panel?
The tool that we have used the most is from IDT, and they have, if you just search for guide RNA design, Cas9, IDT, there's a custom design tool that we use. In the tool, it's just a web-based tool. You can give it a sequence that you want the guide to be placed in, and then it will suggest a number of guides in that sequence. Right now, it's kind of a manual process. You need to give the 1 kb sequence for each place that you want the guide. So you'd have to do that twice, one on either side of the target. But generally speaking, when we use that tool, I would say 75% of the time, the first and the sort of best guide recommended by the tool works well. You will have to test it in the lab. Sometimes you need to redesign and optimize, but that tool has worked very well for us and is what we would recommend.
Thank you for the answers. And then the last question is a little bit different question. Is that using the PureTarget, so like examination using PureTarget, it should be covered by the insurance or can be the X in US?
Yeah, so this is a research use only reagent kit. We have not done any sort of validation by PacBio. We sell the tools as a research use only component of a workflow that can then be validated by customers.